Hello friends, welcome in my channel Pharma Talks. Actually, today's topic is the analytical method development for chromatography. Today, in this series of videos, I will tell the best practice procedure and the perspective behind to start the analytical method development for the liquid chromatography. So what is the perspective behind this? What is the reason and which trend we have to follow, and what are the standard specification during the analytical method development? We are going to discuss all these things in this series of video. So it is a part 1, maybe it should be three to in come last in 3 to 4 parts. So now let's get started. What is the procedure without wasting a time? Okay friends, so in this series of video we can, what is the content in this video, purpose, what is the purpose of the chromatographic development, scope, what is the scope of the chromatographic development, responsibility, who is the responsible for the method development, what is the definition of the method development, procedure, evidence, and last the references. Okay friends, okay, started. Chromatographic development, what is the scope of the chromatographic development? The This best practice document provides detailed information about the analytical method development to be carried out as per ICH guideline. The best practices document is applicable for the analytical method development for drug product. It is meant to drive step by step activities of method development. Okay friends, it is applicable for various test procedures like related substance, assay, content analysis, content uniformity, resolution, residual solvent, etc. Okay friends, so in this video whatever we talk, it is applicable for the both the drug product and drug substances. Who is the responsible for the development of the chromatographic procedure for the liquid chromatography? So research associate or associate scientist, research scientist, senior research scientist, designee is responsible for the analytical method development. Associate scientist, research scientist, senior research scientist, principal scientist, designee is responsible for the reviewing the analytical method development trial and the generated data. Senior research scientist, principal scientist, designee is responsible for the approval of analytical test procedure and analytical method. Okay friends, so what is the definition of the analytical method development? A method development is a set of experimental conditions designed to perform the accurate and precise analysis of a sample as per the requirement. What is the purpose of the method development? To lay down the procedure of analytical method development of a drug product or drug substance using liquid or gas chromatographic technique. So what is the procedure? How we start the method development? It is a very simple. It is the first question arises in the mind who started their method development career. Okay, so what the question started, what is the procedure, how we start the method development? Here is the answer is, method development scientists are expected to have a good understanding of a drug product on or drug substance on which they are working. Okay, so before starting your method development, you have to good understanding of your drug substance or drug product, means which molecule you have to work, you have a complete information about that. Basic knowledge of analytical technique and method validation and transfer, requirement of the respective test and technique. Some of the respected pre-read to actual method developments are. So before starting the method development, you should have to know something, or you should to have to know something. It is a very important for the every method development analyst to start their career in the method development. First you have to know everything about the USP Journal Chapter 621. What is the system suitability parameter as per the USP Journal Chapter 621, and what is the peak count, what is the plate, what is the system, all these things you should be know very well. Along with this, you have to know about the ICS guideline Q2 and Q3 from A to D. In-house analytical method validation SOP, or you should follow the USP journal chapter for the method validation and the method transfer. Okay, patent information leaflet of the drug product and specific guideline if any. So what means, before starting the method development, or you start their career in a method development, so you first know some things. You must know some of the guidelines. You have to read it and you have to learn it, and always remember, and it should be in your mind all time during the method development procedure. Which these things are: the first one is the USP journal chapter for chromatography 621, and the ICS guideline for Q2 Q3. Okay, and the USP journal chapter for the validation and the method transfer, all you should be know very well. So now are going to start at the method development. The first thing, before starting the taking the trial on any column or mobile phase, you should first perform the literature search, because the literature search is the very important technique. With the help of this, you can literally know about the what is your molecule are. So literature search is important in order to collect basic information regarding the physiochemical nature of the drug substance, impurities, and available analytical method. Literature can be collected from in-house data, pharmacopoeia forum, pharmacopoeia, pharma Europa libraries, internet sites like Science Direct, PubMed, Drug Bank, RxList, and various other sources. An overview of material safety data sheet is mandatory, since that content information on the potential hazard, health, fire, reactivity, and environment, and how to work safely with the chemical product. It is an essential starting point for the development of complete health and safety program. So during what should you search during your literature search of your drug product or drug substance? Here are some list. You have to find these properties of your product which can gonna help you during the method development procedure. So the first one is the physiochemical property. The following property can be considered: chemical structure. So before starting the method development, you should compare the chemical structure. In the chemical structure, you should find the presence of chromatographic groups, conjugation, oxochrome, ionic nature, molecular mass of the compound, etc. So before starting the method development, you should know your chemical structure of the molecule. In the chemical structure of the molecule, you should just find out the what what are the presence of the chromophoric group are present in it. Conjugation, is there any conjugation? Is there any oxochrome functional group are present? What is the ionic nature of your molecule? What is the molecular mass of your compound, etc. Physiochemical property of your excipient also you should know before starting the method development. That examples are what type of physiochemical property or excipient you should we know. That is the solubility, chromatographic nature, reactive impurities of excipient, acidity, basicity, swelling, floating property, etc. You should also know about the pH, log P, and pK from the literature or the ACD software, or sometimes you also use the ChemDra for searching of these physiochemical property based on the structure of the molecule. And according to me, 90% of your molecule and their physiochemical property you should be found with the help of ChemDra. Melting point, boiling point, vapor pressure, sample or impurity solubility of analyte in different solvent. So you must search for the solubility or impurity solubility, sample stability and storage. It is very important thing. Sample stability and storage, that should means you should know about the hygroscopicity, photo stability, thermal stability, etc. And the last one is the polymorphism. Okay, You should also know about the polymorphism. So dosage form information. The following factors should be considered: drug product description and number of strength. In case of product with multiple strength, detail like dose proportionate, scale up, down, or pseudo dose proportionate, that is look like, should be taken into account. In case of pseudo pseudo dose proportionate, details of the nature of excipient compensate should be taken into account. Maximum daily dose and allowable impurity limit and genotoxic impurity limit. Packaging and storage information. List of excipient. List of solvent used in DP manufacturing process, API and excipient. So friend, it is the things you should be first know before starting the method development procedure. In my next, the video gonna more long, so I will end this video here, and in the next part we have going to discuss more about the what is the best practices and what should have we have to follow before starting the method development, or how we perform the method development. Okay, for the more videos, and if you want to know more about how we start the method development, watch my part second, method development series part second. Thank you very much.